everyone, welcome to my channel Anupma Biology Classes. In this video, our topic is genetic disorders. This video is taken from CBSE Class 12th, Chapter 5th, Part 5th of Genetic Series. So, the question is, what is genetic disorders? Genetic disorder is a disease that is caused by change or mutation in an individual's DNA sequence. It is an illness caused by changes in a person's DNA. In recent years, a large number of human disorders, deformities, and diseases have been found to be hereditary, in which some of these are caused by gene mutation and some by gross chromosomal abnormalities or mutations. Now, categories of genetic disorders. So, there are various genetic disorders can be grouped into two categories, in which the first one is Mendelian disorders and second is chromosomal disorders. Sometimes there is a mutation, a change in a gene or genes. The mutation changes the gene's instruction for making a protein, does not work properly or is missing entirely. This can cause a medical condition which is known as genetic disorder. Mainly three types of genetic disorders, which are not categories. Categories are only two, but types are three, in which the first one, that is single gene disorder, where a mutation affects one gene, like in case of sickle cell anemia. Second is chromosomal disorder, where chromosomes are missing or changed, like Down syndrome. And the third is complex disorder, where there are mutations in two or more genes. Often your lifestyle and environment also plays a role, like colon cancer. Now, the first genetic disorder, that is Mendelian disorder. So, what is Mendelian disorder? These genetic disorders are mainly caused by alternation and mutation in the single gene. They are transmitted to the offsprings following the principle of inheritance and can be traced in a family by the pedigree analysis. So, there are some common Mendelian disorders are, in which the first one is haemophilia, which is also known as Bridger's disease. So, what is haemophilia? It was first reported by John Otto in 1803. It is defect of the blood which prevents its clotting. Hemophilics bleed excessively when injured, and a seriously affected person may bleed to death after even minor skin cut. It is a sex-linked trait caused by recessive gene, which is denoted by H, located in the X chromosome. This gene controls the synthesis of a protein needed for the clotting of blood. When this gene mutates, it loses its ability to produce the coagulant. Normal blood is dominant to the defective blood. In this picture, you can see that in normal healthy person, clotting occurs after hemorrhage. But in case of hemophilia, after hemorrhage, there is inability to clot, and it happens because of the clotting factor gene present on the X chromosome. So you can see the difference in both cases. That means a healthy person or hemophilic person. Now, after this, inheritance of hemophilia in humans. Man has one X chromosome and one Y chromosome. The gene for hemophilia, being located in the X chromosome, is able to produce the defect because there is no homologue of this gene in the Y chromosome to check its expression. Woman has two X chromosomes. For her to be hemophilic, it is necessary that each X chromosome has a gene for hemophilia. However, such females are usually not born as the combination of two recessive genes is lethal, that is fatal. If only one X chromosome bears a gene for hemophilia, its dominant homologue gene is the other X chromosome will check the expression of the recessive gene, and such females are called carrier. Hence, hemophilia occurs only in the males. Here, there are some conditions Like in first condition which you see in the picture, if the father has hemophilia and the mother is a carrier of the hemophilia gene. So in this condition, in the offsprings, 25% chances of the son will have hemophilia, 25% chance of the daughter will have hemophilia, 25% chances of the daughter will be a carrier of the hemophilic gene, and 25% chances of the son will not have hemophilia. But if father has hemophilia and the mother is not a carrier of the hemophilia gene, in this condition, 0% chance son will have hemophilia and 100% chance that daughters will be a carrier of the hemophilia gene. And if father does not have hemophilia and the mother is a carrier of the hemophilia gene, so in this condition, 25% chances of the son will have hemophilia and 25% chances of the son will be normal. While the 25% chances of the daughter will be carrier of the hemophilia gene and 25% chances that the daughter will be normal. So these all are the conditions of the hemophilia occur in case of the human. Now, the result of marriage of a hemophilic man and a carrier woman. So, if a carrier woman marries a normal man, half of the sons will be hemophilic and half of the daughters will be carrier. The above pattern of heredity is often called criss-cross inheritance because a hemophilic father passes on the gene of hemophilia to his carrier daughters, who then transmit hemophilia to their sons. Such a zigzag pattern of inheritance is typical for all recessive sex-linked traits. Males show the traits, females only transmit them. If a hemophilic man marries a carrier woman, there is a possibility of 50% of the daughters becoming hemophilic. Now, what is the history behind hemophilia? Queen Victoria of England was a carrier and her son Leopold has his defects. Since none of her ancestors and relatives were hemophilic, the mutation probably occurred in the sex cells of her father or mother or in one of her own germ cells. Leopold survived to the father a daughter who was also a carrier. Hemophilia is spread to royal families of Spain and Russia through the marriages of the two of the Victoria's daughter. There is no cure for hemophilia and it may sometimes luckily disappear due to the back mutation. Now, thalassemia. Thalassemia is just like hemophilia and color blindness. Thalassemia also follows Mendelian pattern of inheritance and is caused due to mutation or alteration in a single gene. Thalassemia is an autosomal linked recessive blood disease and transmitted from the parents to the offspring when both the parents are heterozygous carriers for the gene. Here in this picture you can see a normal person blood, that is red blood cells, which is oval in shape, while in case of thalassemia malformed red blood cells form. So, thalassemia differs from sickle cell anemia in that the former is quantitative problem of the synthesizing very few globin molecules, while the latter is qualitative problem of synthesizing wrong functioning globin molecule. Now, the symptoms of thalassemia include anemia, which is caused due to defective or abnormal form of hemoglobin, fatigue, and shortness of breath. Now, in this picture, a black color shows without thalassaemic person, red shows thalassemia, and the half red, half black shows carrier of thalassemia. So there are some conditions in case of thalassemia like when one parent is a carrier. So the risk for child to have thalassemia 0% and become a carrier 50%. While when both parents are a carrier, risk for the child to have thalassemia 25%, become a carrier 50% and 25% unaffected persons. That means child form. While When one parent is a patient, that means thalassemic, and the other is a carrier. So the risk for child to have thalassemia 50% and become a carrier 50%. While when both parents are patient, that means both are thalassemic, so risk for child to have thalassemia 100% and become a carrier 0%. So this is all the cases in case of thalassemic trait and formation of the F1 generations offspring. Now, the second Mendelian disorder, that is the sickle cell anemia. So what is sickle cell anemia? It is an autosomal recessive disease found widely in the South Tropical Africa and also in American blacks whose ancestors came from that part of Africa. It is characterized by sickle-shaped red blood corpuscles formed under low oxygen condition. Here you can see the sickle cell anemia erythrocytes of a healthy person and the human erythrocytes of a patient with the sickle cell anemia. You can see the shape in case of sickle cell anemia, sickle shaped cells are present, while in case of the healthy person, oval shaped cells are present for the RBC. So, changes in the shape of red blood corpuscles is due to the presence in them of a defective type of hemoglobin called sickle cell hemoglobin or hemoglobin S. This is the major effect of the pleiotropic gene and formation of sickle cell corpuscles by the hemoglobin S, that means sickle cell, is one of the secondary effects. The molecules of hemoglobin have a different shape from that of the normal hemoglobin. When the red blood cells containing hemoglobin S face oxygen shortage, their hemoglobin molecules aggregate and form stiff fibers. These fibers distort the cells into odd shape such as sickles. Because of their inflexibility, the sickle-shaped corpuscles cannot easily squeeze through narrow capillaries, unlike the normal flexible corpuscles which change their shape to pass through the narrow capillaries. Because of this, the sickle cell gets stuck in the small capillaries and reduce circulation to the region supplied by their vessels. The sickle cell also break down more rapidly, decreasing the number of red blood corpuscles, leading to anemia, poor circulation, and anemia are other secondary effects of the pleiotropic genes. This effect deprives the tissue of oxygen, and this also produces a variety of other symptoms such as tiredness, headache, fever, muscle cramp, poor growth, jaundice, low resistance to infection, and possibly failure of the heart and kidney. Now, here you can see the chromosome with normal gene and the chromosome with sickle cell gene, which is S in shape. So if a carrier do cross with a carrier, that means one is male and the other is female, so the offsprings will be normal, carrier and affected also. After this, now here you can see the normal hemoglobin gene producing beta chain of the hemoglobin form a normal individual, and in second, sickle cell hemoglobin gene producing beta chain or hemoglobin form an individual with sickle cell. So the gene involved in the sickle cell anemia codes for the beta polypeptide chain of hemoglobin. The oxygen carrying protein present in the red blood corpuscles, imparting them their red color. Sickle cell anemia develops in the individuals who have a sickle allele formed by a change in just one nucleotide pair in its DNA. This alternation results in the substitution of one amino acid, that is valine, from glutamic acid, which is the sixth amino acid in the beta polypeptide chain of the hemoglobin. Now, in this picture you can see the point mutation in case of sickle cell anemia. So, the relevant portion of the beta chain of the hemoglobin where amino acid glutamic acid is substituted by the amino acid valine at the sixth position. This substitution occurs due to the single base substitution at the sixth codon of the beta globin gene from GAG to GUG. This change in the hemoglobin molecules result a change in the shape of RBC from biconcave disc to elongated sickle-like structure. Now, here you can see that inheritance of sickle cell disease. If both parents have sickle cell traits, so what is sickle cell trait? A person have one gene for normal hemoglobin and one gene for sickle cell hemoglobin. That means HbA, HbS. This heterozygous condition is called sickle cell trait. So, if both parents have sickle cell trait, there is a 1 in 4 chance that any given child could be born with a sickle cell anemia. And there is also a 1 in 4 chance that any given child could be completely unaffected. And there is a 1 in 2, that means 50% chance that any given child will get the sickle cell trait also. Here you can see a marriage between the two carriers produces affected, that means diseased, carriers and normal, that is disease free children in the ratio as shown in the figure. The affected individuals die when adult. So when we cross two sickle cell trait, then the offspring's normal 25%, sickle cell trait 50% and the sickle cell anemia 25%. Now, inheritance of advantages of sickle alleles. So, what are the advantages of sickle alleles? Some 20 to 40% of the people in tropical Africa have the sickle allele in heterozygous condition. This shows that the heterozygotes have some selective advantages. Presence of a sickle cell allele lowers the heterozygotes' chances of developing malaria. The RBCs having abnormal hemoglobin sickle more readily when they are infected with the malarial parasite. The parasites of sickle cells die. The remaining parasites are then destroyed by the body's defense before malaria develops. Thus, the sickle allele in heterozygous condition protects the person against malaria. Now, how to avoid sickle cell affected individuals? So, birth of a children with fatal sickle cell anemia can be avoided by discouraging marriages among the heterozygotes. The heterozygotes can be identified by microscopic examination of their blood. After this, now the third Mendelian disorder, that is Allopolyploid. So what is Allopolyploid? It is an inborn metabolic disorder produced by the action of a single gene. Now, here you can see the symptoms and sign of Allopolyploid like black colored urine, onchronosis, tendinitis, kidney and prostate stones, kyphosis or hunchback. So the symptoms include blackening of urine on exposure to air and darkening of certain cartilages. Urine of alkaptonuric person contains homogenistic acid, also called alkapton, which spontaneously combines with the oxygen to form a black pigment, and this pigment slowly deposits in the cartilages of the ears and nose. It also causes mild arthritis. Now, what are the causes of the alkaptonuria? So here you can see that in normal persons, a liver enzyme homogeneous oxidase catalyzes the breakdown of homogenistic acid to compounds that enters Krebs cycle to produce carbon dioxide and water, which you can see in the picture. The alkaptonurics are unable to break down homogenistic acid because of the absence or inactivity of the enzyme needed for this purpose. Lack of the enzyme is due to the absence of the normal form of the gene that controls the synthesis of the enzyme, Hence, homogenistic acid accumulates and is excreted in the urine. Now, fourth is phenylketonuria. In short, you can say PKU. So, what is phenylketonuria? It is a recessively inherited disorder and also an example of pleiotropy. It causes extreme mental retardation in children. It also results in hyperpigmentation of skin and eczema. It occurs in about 1 out of every 10,000 births in United States. The affected baby is normal at birth, but within a few weeks, phenylalanine level in plasma starts rising, and by the age of 6 months, he develops severe mental retardation. If deficiency of phenylalanine hydroxylase is detected in the newborn, disorder can be usually prevented with a special diet low in phenylalanine. So, what are the causes of the phenylketonuria? Here you can see two conditions. First one is normal, where the tyrosine form, and the second is phenylketonuric condition, where the tyrosine is not formed because of an enzyme which is known as phenylalanine hydroxylase. So, the homozygous recessive individuals lack the enzyme phenylalanine hydroxylase needed to change one amino acid, phenylalanine, to another tyrosine. Phenylalanine accumulates in the tissues and some of it changes into phenylpyruvic acid. The latter is excreted in the urine. Accumulation of phenylalanine and its metabolites damage the brain and causes the disease. Lack of enzyme is due to the abnormal recessive gene. The heterozygous individual is normal but carrier. So, this is the condition of phenylketonuria. After this, now the sixth is albinism. Here you can see what the causes and symptoms of albinism. It is a recessively inherited disorder. What are the symptoms? It is characterized by the lack of dark pigment melanin in the skin, hair and iris. It by itself is not a disability. However, an albino is susceptible to eye disorders due to the damaging effect of bright light. What are the causes of albinism? It is caused by the absence of enzyme tyrosinase, which is necessary for the synthesis of pigment melanin from dihydroxyl phenylalanine. The gene for albinism, that is small a, does not produce the enzyme tyrosinase, but its normal allele A does. Thus, only homozygous individual, that is small a is affected. So this is all about albinism. Now, the next is red-green color blindness. So what is red-green color blindness? In this picture you can see the red-green color blindness is also a sex-linked recessive trait. And here the inability of a certain human beings to distinguish red from green color. Red-green color blindness is produced by an X-linked recessive gene by causing lack of one of the primary cone pigments of the retina. Normal eyesight is dominant to color blindness. After this, here you can see the inheritance in case of the red-green color blindness. Male has one X chromosome and one Y chromosome. The gene for color blindness lies in the X chromosome and is able to produce the disorder because there is no homologous gene in the Y chromosome to check its expression. Female has two X chromosomes. For a female to be colorblind, it is necessary that each chromosome has a gene for color blindness. If only one X chromosome bears a gene for color blindness, its dominant homologous gene on the other X chromosome will check the expression of the recessive gene. Hence, color blindness occurs more often in the males than in the females. So here, if an unaffected female carrier cross with the unaffected male, then in the offspring there are four offsprings: unaffected female 25%, unaffected female, that means carrier which is also 25%, unaffected male which is non-carrier 25%, and the colorblind male and carrier which is 25%. So this is a genetic diagram for the inheritance of color blindness. Now here in this picture you can see the inheritance pattern in the color blindness. A colorblind man and a normal woman, which is not a colorblind, not a carrier, produces normal sons and carrier daughters. Here, no colorblind sons and all daughters, that means 100% chances of the colorblind carriers. And if a colorblind female, that means mother, cross with a normal man, in this condition 100% carrier, means all the daughters are carrier of the color blindness, and a 100% color blind son will be formed or appear in the next generation. Now next is night blindness. So what is night blindness? It is of two types, acquired and congenital. The acquired night blindness is due to the vitamin A deficiency. The congenital night blindness is due to visual purple deficiency that interferes with the function of the retinal rods. It is a sex-linked trait. It is caused by a recessive gene carried by the X chromosome. Like other sex-linked traits, night blindness is also more common in males than in females. It is inherited like red-green color blindness. Man suffers from night blindness even by having a single gene for the defect because there is no homologue of this gene in the Y chromosome to check its expression. Females become night blind only if she receives two genes for the defect. Here you can see all the symbols to detect here that in first picture you can see all the inheritance pattern in case of night blindness or color blindness, both cases. If a male is normal and the female is carrier, in this condition 50% chance of son having abnormal color vision, while the 50% chances of the daughters being carrier. In case if a abnormal male crosses with the normal female, in this condition 100% chance of the son having normal color vision and the 100% chances of the daughters being carrier. While if the male is abnormal and the female is carrier, in this condition 50% chances of the son being colorblind and the 50% chances of the daughter being colorblind or being carrier. And if the male is normal and the female is colorblind, in this condition 100% chances of the son being colorblind and the 100% chances of the daughter being carrier. So these all are the inheritance pattern in case of color blind or night blindness. Now next is muscular dystrophy. What is muscular dystrophy? It causes deterioration of muscles at an early age. The mutated gene on X chromosome fails to produce a protein called dystrophin. This protein is thought to relay the nerve signals to the calcium storage sacs in the muscle cells. As a result, calcium is not released and the muscle contraction is halted at the very first step. Further, abnormal calcium level triggers the release of an enzyme that destroys actin and myosin. The victim becomes invalid by the age of 10 and usually dies by the age of 20. So these all are about the muscular dystrophy. Here in this picture you can see a single inheritance pattern in case of the muscular dystrophy. Here healthy father crosses with the healthy mother but the mother is carrier. In this condition the offspring will be healthy son 25%, healthy daughter which is non-carrier 25%, healthy daughter which is carrier 25%, and the son with Duchenne muscular dystrophy, that is DMD, is 25%. So this is the case of muscular dystrophy. Now the second genetic disorder, that is the chromosomal disorder. So what are the chromosomal disorder? Chromosomal disorders are caused due to absence or excess or abnormal arrangement of one or more chromosomes. Some examples of the chromosomal disorders are in which the first one is aneuploidy. So what is aneuploidy? It involves the addition or deletion of one or a few chromosomes from the usual diploid set of chromosomes. Sometimes the two homologous chromosomes of a particular pair fail to separate in meiosis first, or two sister chromatids fail to separate in meiosis second. This is called non-disjunction and its result in that one gamete receives both the chromosomes of a pair and the other gamete is left without that particular chromosome. Now, here you can see that is non-disjunction in case of aneuploidy. So, an egg with two homologous chromosomes fertilized by the sperm with one homologous chromosome, maybe X or Y, will form an individual with three homologous chromosomes, that is XXX condition or XXY in this case. The XXY individual is sterile male with many female features. Union of an egg without an X chromosome with an X carrying sperm produces an individual having a single X chromosome. This is sterile female will with many male features. So the presence of a single copy or three copies of a particular chromosome, that is X or triple X, is called monosomy 2n-1 condition or trisomy 2n+1 condition. An aneuploid having four copies of one chromosome is known as tetrasomy, that is 2n+2 condition. And trisomy and tetrasomy are thought to have produced new species such as apple and pear by secondary polyploidy. An aneuploid lacking a pair of homologous chromosomes is said to be nullisomic, that is 2n-2 condition. So, aneuploidy is of two types. One involving loss of chromosomes is called hypoploidy, that is 2n-1, which you can see in this picture. And that causing addition of chromosome is termed hyperploidy. That means 2n+1 condition. The organisms showing aneuploidy are called aneuploids or heteroploids. Aneuploidy may also arise by a loss of some chromosomes from the normal or polyploidy number, thought faulty mitosis. So this is the case of fault, that means mitotic mutations, like the non-disjunctions of the homologous chromosome meiosis first and the non-disjunction of sister chromatids in meiosis second, which you can see in the picture. And this non-disjunction is responsible for formation of aneuploidy and polyploidy. Now, the next second is polyploidy. So what is polyploidy? It is the addition of one or more complete set of chromosomes or genomes. It is also called genomic mutation. In general, the polyploids are more nearly normal in appearance than aneuploids. One extra or missing chromosomes apparently disrupts genetic balance more than does an additional full set of chromosomes. Polyploidy is of two types, in which the first one is autopolyploidy. It arises by increase in the number of chromosomes set of the same species. It is thus an intraspecific polyploid. It is rare in nature. It is easily induced by the temperature variations, radiations, and chemicals such as colchicine and granosine. Here in this condition, you can see the examples of autopolyploids like the plant, that is potato, which have 4n polyploid and chromosome number is 48. In case of banana, 3n condition, that means 33 chromosomes number. In peanut, 4n condition, that is 40 chromosomes number. And in the sweet potato, 6n condition, that is 90 chromosome number. So, a Diploid species having two similar chromosome sets, that is AA, will change into an autotriploid, that means AAA, by duplication of the chromosomes of one set, and into an autotetraploid by the duplication of the chromosomes of both the sets. So the autopolyploids have sets of the same genome. The autotriploid is sterile because the chromosome of its third set have no homologous to pair within meiosis, and it can be propagated asexually. So the seedless watermelon produced by H. Kihara in Japan is autotriploid, and the seedless grapes and banana are autopolyploids. So Autopolyploids is rare in nature. Here you can see that is karyotype of parent species, that is 2n equal to 6. In meiotic error, unreduced gametes with 6 chromosome forms. And after self-fertilization, the zygote is formed autopolyploid, that is 4n equal to 12. It means tetraploid condition. Offspring with polyploid karyotypes may be viable and self-fertile. Here you can see the example, that is the triticale. How can it form? It is a case of autopolyploid. Here the triticum durum, that is tetraploidy condition, and the sickle serial, that is diploidy condition. Then the gametes form, that is 14 in case of triticum and 7 in case of sickle, because half of the number of chromosomes in case of gametes. Then in F1 hybrid, sterile offspring, that is triploidy conditions form. And here the chromosome doubling by colchicine form 42 chromosome, that is known as triticale. It means combination of triticum and sickle, and this is a hexaploidy condition. Now the second type of the polyploidy is allopolyploid. It develops in a hybrid between the two species by doubling of chromosome sets, that is AB to AABB. It is thus an interspecific polyploidy. The allopolyploids have multiple sets of two different genomes. Now, here you can see that allopolyploidy speciation. How that it forms. Here the species A and species B. Species A have 4 chromosomes and species B have 6. In species A, because of the meiotic error, chromosomes number not reduced from 2n to n. So unreduced gamete with 4 chromosomes, and in species B, it's normal gamete, that is 3 chromosomes. Then the hybrid with same n chromosomes forms after the fertilization in step first. Then the unreduced gametes with 7 chromosomes crossed with normal gametes of the species B and form the viable fertile hybrid, that is known as allopolyploid, that is 2n equal to 10. So the allopolyploid contains the sum total the chromosomes of the two parental species. This is known as allopolyploid speciation. Now, in this picture, you can see an example, classic example of the allopolyploid, which is a new plant species, that is Raphanobrezica, which is produced by a Russian geneticist G.D. Karpenchko in 1927. He crossed two plants of different genera, the radish, that is Raphanus sativus, and the cabbage, that is Brassica oleracea, each of which has 9 pairs of chromosomes. That means 18 chromosomes. The hybrid produced were diploid, having 9 radish and 9 cabbage chromosomes. All hybrids except a few were sterile because the chromosome of the different plant genera could not pair in meiosis. In some sterile hybrids, doubling of chromosome occurred, forming 36 chromosomes, 9 pairs of radish and 9 pairs of cabbage, and these plants then breed true because of the 36 chromosome could form 18 pairs. So this is known as a allopolyploid condition in case of Raphanobrezica. Now, the next is here, that is the Down syndrome. So what is Down syndrome? It is called Mongolian idiocy, that is Mongolism. It was first reported in 1866 by Langdon Down, but its cause was found in 1959 by Lejeune and his co-workers. So, if you talk about the symptoms of the Down syndrome is characterized by rounded face, broad forehead, a Mongolian type of eyelid fold, flattened nasal bridge, permanently open mouth, projecting lower lip, protruding tongue, short neck, flat hand, and stubby fingers. The victim has little intelligence due to malformation of the brain. Heart and other organs may also have deformities. Gonads and genitalia are underdeveloped. Few victims live in maturity, but those who attain maturity produces Mongolian and normal children in about equal proportion. So this is the symptoms of the Down syndrome. Now here, the cause of the Down syndrome, that is case of the trisomy. Okay, so Down syndrome is an aneuploid trisomic disease. It is caused by the presence of an extra chromosome number 21, and both the chromosomes of the pair 21 pass into a single egg due to non-disjunction during oogenesis in the mother's ovary. Thus, the egg has 24 chromosomes instead of 23, and the offspring has 47 chromosomes. That means 45 plus XY in male and 45 plus XX in case of female. Instead of 46 chromosomes, so the presence of three chromosomes of the same kind is called trisomy, and the Down syndrome is also known as 21st trisomy. The incidence of non-disjunction rises in the ovaries of aged women. Therefore, the women above 40 years of the age are more likely to produce children having Down syndrome. They, if pregnant, can have fetal testing for trisomy 21. One in every 600 children may have Down syndrome. Individuals having Down syndrome are likely to develop leukemia and Alzheimer's disease. So this is the case of non-disjunction of cell division in trisomy 21st condition in Down syndrome. Now, the familial Down syndrome. What is familial Down syndrome? This syndrome may run in the family, hence its name. It arises by translocation of a large segment of or whole of the third chromosome 21 to another chromosome, usually 14. In some cases, the total number of chromosomes remains normal, that is 46, but there is an extra chromosome 21 material. This form of the Down syndrome is rare. The small extra chromosome carries enough excess genetic information to disrupt many processes in the body. So these all are about the Down syndrome. Now the next is Klinefelter syndrome. So what is Klinefelter syndrome? It is an aneuploid condition with three sex chromosomes, that is trisomy. Here you can see that is cause of the Klinefelter syndrome. Klinefelter syndrome is caused by XXY genotype. This genotype results from the union of a non-disjunct XX egg with a normal Y sperm or normal X egg and abnormal XY sperm. The individual have 47 chromosomes, that is 2n plus 1 condition. Here in the picture you can see the arrow indicates that is XXY condition. It can be formed by the normal XX egg with a normal XX egg which is non-disjunct with the Y sperm or normal X egg with the abnormal XY condition. So what are the symptoms of the Klinefelter syndrome? A person having Klinefelter syndrome is a sterile male with a small testis, usually long legs, obesity and sparse body hair, and with many female characteristics such as breast. The victim usually has normal intelligence. The more the X chromosomes, the greater is the mental defect. Klinefelter syndromes occurs about once in 2000 live birth. So these are all about the Klinefelter syndrome. Now the next is Turner syndrome. So what is the Turner syndrome? It is an aneuploid condition with a single sex chromosome, that is monosomy condition. It is caused by exogenotype. This genotype results from the union of an abnormal O egg with a normal X sperm or a normal X egg and abnormal O sperm. The individual has 45 chromosomes, that is 2n minus 1 Turner syndromes, the only known viable monosomy in case of humans. Now, here are the symptoms. The individual having Turner syndrome is a sterile female with underdeveloped breast, reduced ovaries, a small uterus, loose skin of the neck, mostly normal intelligence and short stature, and with many male characteristics such as heavy neck muscles and narrow hips. She may not menstruate or ovulate. The incidence of Turner syndrome is 1 in 5,000 births, and the combination is Y or sex chromosome is not viable. It means Y carrying a sperm plus O egg is not viable. So because of this, after this, what are the differences in the Klinefelter syndrome and Turner syndrome? So according to the picture, you can distinguish, but the Klinefelter syndrome is caused by the genotype 44 plus XXY condition, while the Turner syndrome is caused by the genotype 44 plus XO condition. Klinefelter syndrome, in which the individual is a sterile male, while in case of Turner syndrome, individual is a sterile female. Klinefelter syndromes, individuals have small testes, long legs, sparse body hair, while in Turner syndrome, individuals have reduced ovaries, short stature, a small uterus, undeveloped breast, and loose neck skins. In Klinefelter syndrome has some female characters such as enlarged breast, while in Turner syndrome some male characters such as heavy neck muscles and narrow hips. It means the Klinefelter syndrome and the Turner syndrome both are antagonistic to each other. After this, now the next is Patau syndrome. What is this? Fewer than 100,000 cases per year. It causes severe intellectual disability and the physical defects. It is a serious rare genetic disorder caused by having an additional copy of chromosome 13 in some or all of the body cell. Surgery may be necessary to repair heart defects or cleft lip and cleft palate. Physical, occupational, and speech therapy will help individuals with Patau syndrome reach their full developmental potential. In this picture, you can see there is Patau syndrome, that is trisomy 13. In this case, kidney, heart, CNS malformation occur like the Edward. Polydactyly, 80% die within first year, can be diagnosed in utero via ultrasound and the maternal serum markers like alpha fetoprotein. Here in this picture you can see the sign and symptoms like the small head, absent eyebrows, cleft lip, palate, and dysplastic or malformed ears, clenched hand and polydactyly or extra fingers, undescendant or abnormal testes. So these are the symptoms of the Patau syndrome. Now, the next is Edward syndrome. What are the causes and what is Edward syndrome? It is trisomy 18. It is a chromosomal condition associated with abnormalities in the many parts of the body. The average lifespan for the infants born with trisomy 18 is 3 days to 2 weeks. It is rare, occurring in about 1 in 2500 pregnancies, and this rare syndrome is 3 times more common in females rather than males. In case of Edward syndrome, here you can see the symptoms like the congenital heart defects, growth retardation, dysmorphic features like the small mouth, small jaw, short neck, shield chest or short and prominent sternum and wide set nipples, flexed big toe, prominent heels, clenched hands with overlapping fingers, dysplastic or malformed ears, occiput or back part of the skull is prominent, facial cleft, spina bifida, several developmental delay occur in this case. And in Edward syndrome, it is 18 trisomy, kidneys, heart, CNS malformation occur, many dies in uterus, half of those born don't survive past first week, can be diagnosed in utero via ultrasound and maternal serum markers like alpha fetoprotein. So this is all about the Edwards syndrome. Now, after this, what are super males and super females? So, in case of super females, these have genotype XXX, XXXX or 5 times X with total chromosomes number 47, 48 or 49. Such females show abnormal sexual development and mental retardation. These symptoms become more severe with the increasing number of X chromosomes. Now, what are super males? These have genotype XYY condition. They have unusual height, mental retardation, and perhaps criminal bent of mind. They are more aggressive than normal males and usually overproduce male hormones. After this, now the human genetics. So, the branch of genetics which deals with the inheritance of characters in humans is called human genetics. The foundation of the human genetics was led by Sir Archibald Garrard, a British physician, in 1902 by pointing out that the inborn errors of the metabolism are controlled by genes and these errors are inherited in a Mendelian fashion. It is known as father of human genetics. Human hereditary traits. What are human hereditary traits? About 200 traits are transmitted from generation to generation in humans, and these are called hereditary traits. These include dominant as well as recessive, and many human traits are transmitted in a simple Mendelian manner, others in accordance with the post Mendelian principle of genetics. So, here you can see the some main human hereditary traits. So, like characters here, which is dominant is dark, curly, and recessive is light, straight. Skin color normal is dominant, and albino is recessive. Eyes are brown, large, dominant, and blue is small, recessive. Eyelashes, if long, then dominant, if short, then recessive. Color vision is normal, dominant, and color blindness recessive. Lips are thick then dominant and thin then recessive. Nose are big then dominant, small then recessive. Nostrils are wide then dominant and narrow then recessive. Hearing and speech is normal then dominant and deaf and mutism is recessive. Blood clotting, if normal, then dominant, and haemophilia, then recessive. RH factor is positive, then dominant, and negative, then recessive. And ear is long, wide, free, then dominant, and short, narrow, fused is recessive. So these all are the human hereditary characters or traits which are, you can see here, that is dominant and recessive, and these are show the Mendelian pattern of inheritance. Now, the methods of study, in which the first one is the pedigree record. So, the five basic approach are followed these days to study human genetics. In case of pedigree record, proper pedigree records of the families are maintained, and this help in tracking the transmission of a particular trait through generations. Second is population genetics. The problem faced by geneticists on account of a small number of children in the human family have been overcome by methods devised to analyze the fate of characters in populations. So, population genetics, a new branch of biology, is very helpful in the study of human genetics. It involves the study of distribution of traits and estimation of gene frequency in a population of organisms. The autosomal dominant recessive inheritance in a population can be expressed by Hardy-Wingner law or equation. They independently proposed in 1908 that genotype and phenotype frequencies could be determined by a simple algebraic expression, which is p square plus 2pq plus q square equal to 1, where the p square represents homozygous dominant individuals, q square represents homozygous recessive individuals, and 2pq represents heterozygous individuals. Now, algebraic expressions have their meanings. Like the p plus q equal to 1, it means all dominant alleles plus all recessive alleles equal to all the number of a particular gene in a population. P square plus 2pq plus q square equal to 1 means for a particular gene, all homozygous dominant individual plus all heterozygous individuals plus all homozygous recessive individuals are equal to all the individuals in the population. The normal frequency of a dominant allele is 2 by 3 and that of a recessive allele is 1 by 3. So, this equation is analogs to the monohybrid cross, that is capital B small b into capital B small b, which yields homozygote, that is capital B and small b and the heterozygotes capital B and small b capital B. Or capital B plus 2 capital B plus small b. It represents the P square plus 2pq plus q square equal to 1. Now, the human cell culture. The techniques of cultivating human cells in a specific media and the techniques of somatic cell genetics have enabled geneticists to understand the biochemical basis of the inheritance of many characters. Next is human cytology. The correct number of somatic chromosomes in man was not known till 1956. New techniques of staining chromosomes have revealed their correct number, and this has greatly helped the study of human genetics. And the last is study of twins. The identical twins develop from a single zygote by separation of the two blastomeres after the first cleavage. Therefore, they have similar genetic traits except for the rare change of mutations. Fraternal twins develop from different zygotes. Therefore, they are no more alike than other children born at different times in the same family. Since the identical twins carry the same genotype, their study help in distinguishing between the hereditary and the environmental characters. The traits that are similar in the identical twins are considered hereditary, and the features in which they differ are regarded environmental. Such studies have shown that intelligence, diabetes mellitus, feeble-mindedness, and many others are hereditary traits in case of human or man. So these are the methods of study of the human genetics. Now, human chromosomes. So first, that is the number. The normal diploid chromosomes in human being is 46. These includes 22 pairs of autosomes and 1 pair of sex chromosome. A normal female has 22 pair of autosomes and an XX pair of sex chromosome. A normal human male has 22 pairs of autosomes and an XY pair of sex chromosome. Each chromosome pair has a distinct morphology as regard the size, shape, position of the centromere, relative length of the arms and presence of the satellite. On this basis, it can be distinguished from other pair of the chromosomes. Second is karyotype. It is the systematized representation of the chromosome complement of a cell or organism depicting the characteristics of the chromosome as seen in the metaphase of mitosis. The photographs or diagrams of the chromosomes seen in a single cell nucleus are artificially arranged in the homologous pair, which are arranged in order of their size and according to centromere position, except the sex chromosome that are placed at the end. So this is the end of lecture in which you knew about the genetic disorders, in which some are Mendelian and some are chromosomal disorders. After this, human genetics and the human chromosome also. So the next upcoming video is molecular basis of inheritance, and this is the first video of the molecular biology. So if you understand this video, please like and share it and subscribe to my channel Anupma Biology Classes. If you have any questions, any queries or any suggestions, you can ask in the comment section below. Thank you for the watching.